Hello students, I am Dr. Neela Mahajan. I shall be discussing good manufacturing practices part 1. In this module, we will introduce ourselves to the concept of good manufacturing practices, consequences of GMP compliance, GMP guidelines, certification, then personnel, equipment and building concern, control of production records and procedures, and elements of manufacturing control. Good manufacturing practices or GMP is a quality assurance exercise which aims at the control and management of manufacturing, testing and overall quality control of products. It ensures consistent production and control of products as per quality standards appropriate to their intended use and marketing authorization requirements. Occasionally called as CGMP or the current methodologies of manufacture, testing, design and control, CGMP regulations are applicable to OTC as well as prescription drug products. There are various consequences of GMP compliance which include the saving of the firms from serious non-compliance consequences like recall, seizure, fines and jail. It diminishes risk inherent in any pharmaceutical production like cross-contamination and mix-ups. Interest of the consumer from purchasing an ineffective or dangerous product is safeguarded by GMP compliance. Now the GMP guidelines. World Health Organization or WHO guidelines are the minimum basic standard to be attained by any country locally manufacturing pharmaceuticals. The latest version recognizes a number of major national and international reference texts to GMP. Moreover, it acknowledges the role of ISO 9000 series of standards in the management of quality. Practically, these are only applicable to countries that do not intend to export their products widely. These are not accepted as an appropriate standard for imports. WHO guidelines are published in 3 parts. Part 1 consists of general approach to quality assurance and main elements of a GMP system. Part 2 comprises of review of the separate responsibilities for production and QC personnel to comply with GMP requirements. Part 3 comprises of the annexes or supplementary guidelines for manufacturing of sterile pharmaceutical products and GMP for active pharmaceutical ingredients. It is intended that further annexes would be added with time. Under GMP, all manufacturing processes are clearly defined, systematically reviewed in the light of experience and are capable of consistently manufacturing pharmaceutical products of the required quality compliant with their specifications. Qualification and validation are performed and all necessary resources are provided which include appropriately qualified and trained personnel, appropriate materials, containers and labels, suitable equipments and services, approved procedures and instructions, suitable storage and transport, adequate premises and space, and lastly adequate personnel, laboratories and equipment for in-process controls. Under GMP, instructions and procedures are written in clear and unambiguous language specifically applicable to the facilities provided. Also, operators are trained to carry out procedures correctly. Moreover, records are made during manufacture as a proof that all the steps as per the defined procedures and instructions have in fact been taken and the actual quantity and quality matches the expected product quantity and quality. All significant deviations are fully recorded and investigated. Moreover, under GMP, comprehensible records covering manufacture and distribution are retained in an accessible form to enable the complete batch history to be traced. Under GMP, proper storage and distribution of the products minimizes any risk to their quality, and a system is available to recall any batch of product from sale or supply. Moreover, complaints about marketed products are examined, causes of quality defects investigated, and appropriate measures are taken in respect of defective products to prevent recurrence. There are many different versions of GMP in use around the world with the common objective. Now the GMP certification. GMP certification of an organization is an authorization and certification of company's product and process that quality standards are adequate, up to date and controlled for end use by the consumer. Steps for GMP implementation are defining standards, training, establishing system, documentation and record, audit, review and correction, and finally certification. Let us summarize the benefits of GMP requirements. The first benefit of GMP is it is a good business tool for refining compliance as well as performance of the firm. It gives freedom to manufacturers to select the best possible way to implement the necessary controls. It aims at continuous improvement. It is general, open-ended, and shifts focus of companies to a quality approach. And last but not the least, it is largely common sense based. Now we will discuss the elements of GMP one by one. First of all, we will discuss personnel. Proper selection, training, and motivation of personnel are vital for consistent production of quality pharmaceuticals. Quality consciousness in the personnel infused by proper direction and motivation minimizes the risk of errors in production of quality pharmaceuticals, with focus on prevention of the mistakes rather than their detection. Key personnel involved in the manufacture and control of the quality drug are responsible for assuring that drug and dosage form being handled have the desired characteristics. Therefore, it is essential to employ qualified personnel to supervise formulation, processing, sampling, testing, packaging, and labeling of the drug. Responsibility for keeping the manufacturing process within the CGMP regulations must be delegated. People directly involved with various aspects of the process and their immediate supervisors are the right persons. Supervisors should direct and control the operation and should be available at all times. Competent staff should be placed in charge of the maintenance of machinery, equipment, and sanitation. Operating personnel should be authorized to sign the manufacturing documents for all the process for which they are accountable. The document is then countersigned by the supervisor. Signature and endorsement should appear on the proper worksheet at the completion of a production operation. Now we will discuss equipment and buildings under GMP. Equipment and buildings used in the manufacture, processing, packaging, labeling, storage, or control of drugs should be of suitable design, size, construction, and location. Further, these should be maintained in a clean and orderly manner. The building should provide adequate space for the orderly placement of materials and equipment, should minimize any risk of mix-ups or cross-contamination between the drugs, excipients, packaging, and labeling, should provide adequate lighting, ventilation, dust control, temperature, and humidity. It should make provisions favorable to product integrity and safety, such as bacteriological controls for sanitizing work areas for parentals and for prevention of the dissemination of microorganisms. The desired characteristics of equipment for producing quality products are numerous and differ from machine to machine. However, the equipment should be of suitable size, accuracy, and reproducibility. It should have inert, non-reactive, non-absorptive, and non-additive surfaces. It should be constructed to facilitate adjustment, cleaning, and maintenance. This in turn assures precision, reliability, and uniformity of process as well as product. Now we will discuss control of records under GMP. Records like master formula and batch production records should be prepared and maintained in accordance with the established procedures. Master formula record. Information in the master formula record should be provided in a simple, understandable format and language. This not only reduces the risk of misinterpretation of information by the operating personnel and the supervisor, but also assures identical reproduction of each batch of a product. Master formula records for each product should be prepared, endorsed and dated by a competent responsible individual and should be independently checked, endorsed and dated by another competent and responsible individual. Though product-wise content and format may vary, master formula record should include certain information which include the name of the product, a description of the dosage form and its strength, complete list of ingredients designated by whole names and codes sufficiently specific to indicate any special characteristic, quantity by weight or volume of each ingredient regardless of whether it appears in the finished product. If variations in the quantity of a particular ingredient are permitted, as is sometimes necessary in the preparation of a dosage form, an adequate statement should be provided in the record. The standards or specifications of each ingredient used in the product. It should also include an appropriate statement concerning any calculated excess of an ingredient. Further, master formula record should also have appropriate elements of theoretic yield at various stages and the termination of processing. It should also have manufacturing and control instructions, specifications, precautions and special notations to be followed. Last but not the least, it should have a detailed description of closures, containers, labeling, packaging and other finishing materials. Now the batch production record. This record shall contain an accurate reproduction of the manufacturing formula, procedure and product specifications from the correct master formula procedure to be used in the production of a batch. These batch records are then sent to each of the departments involved in the production, packaging and control of the product. Batch production records should be prepared, maintained and controlled for each batch of product. In general, they should be retained for a period of approximately 5 years after distribution has been completed. The records include dates, specific code or identification numbers of each ingredient used, weights or measures of components and products in the course of processing, results of in-process and control testing, endorsements of the individual performing and supervising each step of the operation, and a lot number for the identification of all the procedures performed on the lot and their results. This lot number appears on the label of the product and helps to search for details of manufacture and control history of any particular product. Now control of production procedures under GMP. Under GMP, production and related in-process quality control procedure, that is IPQC, is a planned system for identification of materials, equipment, processes and operators. It is also used for enforcing the flow of manufacturing and packaging operations as per the set rules and practices and for minimizing human error or to detect the error if and when it does occur and fixing the responsibility to the personnel involved in each unit operation of the entire process. It should be rigidly followed as required by the master formula record or the batch production record. Detects variations from the tolerance limits of the product followed by prompt and corrective action. It leads to products having intended characteristics of identity, strength, quality and purity. It includes rapid and simple tests or inspections performed during manufacturing of a product batch. It provides accurate, specific and definite descriptions of procedures to be used from the receipt of raw materials to the release of the finished dosage forms. The IPQC procedures and tests should be openly discussed, experimentally justified, written in detail, properly explained and after being established, rigidly enforced. Now we will discuss elements of manufacturing control under GMP. The primary objective of an IPQC system is to monitor all the features of a product that may affect its quality and to prevent errors during processing. Its elements are common. The production planning department issues the formula and manufacturing worksheets. These worksheets bear an identification number and the names and quantities for all ingredients, a complete description of the procedures to be followed, precautions to be taken, and equipment to be used, lot sizes to be processed, and the suitable in-process controls to be undertaken. Production department writes and issues material tickets for each raw material to the Department of Material Stores where orders are filled and verified. After materials have been sent to the production department and checked similarly, the pattern of control takes shape as production proceeds. A qualified person verifies and countersigns the addition of raw materials to the batch. Notation of the identifying number of each ingredient is made on the manufacturing worksheet as it is used, and each unit operation is checked off as it is performed. An appropriate label is attached to each container or piece of equipment in use to identify its contents and to ensure that in-process stage is properly designated. Product quality audit is done at various stages of production. In-process checking is done by auditor or the control inspector who checks, enforces and reviews procedures and suggests the change for upgrading the procedures when needed. Every smallest deviation from standard operating conditions should be reported to concerned production and control personnel. The in-process quality controls for parenteral products include subjecting the product to physical examination for appearance, clarity, and particulate contamination, examining the sterility indicator placed in various areas of sterilizer for each sterilization operation, checking the bulk solution before filling for drug content, pH, color, clarity, and completeness of the solution, checking the filled volume of liquids or filled weight of sterile powders for injection in the final containers at predetermined intervals during filling, and testing for leakage of flame sealed ampoules, and submitting the product for sterility testing or other predetermined biological tests to establish the safety and other parameters of the product. Now the in-process quality control for solids. It includes determining the drug content of the formulation, checking the weight variation for tablets and capsules at predetermined intervals during manufacturing, checking the disintegration and dissolution time, hardness, and friability of tablets at least during the beginning, middle, and end of production or at prescribed intervals during manufacturing, testing soluble tablets for compliance with solution time requirement, examining products by line inspection or other equally suitable means, and removing the defective units prior to packaging, and determining drug content of formulation. The in-process quality control for semi-solids includes checking for uniformity and homogeneity of drug content prior to filling operations, determining the particle size of the preparation when appropriate, checking the appearance, viscosity, specific gravity, sediment volume, and other physical parameters at prescribed intervals, testing for filling weight during the filling operation, and testing for leakage on the finished jars or tubes. After the in-process quality control, at the completion of the manufacturing process as well as in-process stages, actual yields are checked against theoretic value and the representative samples are withdrawn for laboratory testing by the control inspector as per predetermined sampling plan. The operators actively performing the process, their supervisors and control inspector must all verify that entire operation was accomplished in the prescribed manner. Occasionally, materials in bulk storage are randomly sampled and examined. Absence of detectable changes confirm that the batch is satisfactory for final packaging. The batch production records and other needed documents are then delivered to the quality control office together with the withdrawn samples of the product. These records and test results are reviewed for conformance to specifications and current GMP. The bulk finished products are held in quarantine until they are released for packaging by quality control personnel. Now, packaging control under GMP. Packaging control comprises of certain steps which we are going to discuss. Before the manufacture of a product is completed, a packaging record is issued to the packaging section. This record bears an identification number and specifies packaging materials to be used, operations to be performed, and quantity to be packaged. Simultaneously, requisitions placed for the products to be packaged. The packaging and printed materials like labels, containers, inserts, brochures, cartons, and shipping cases. The packaging operation unites the product, container, and label to form a single finished unit. Individual package components must be correct and assembled correctly. Prior to starting a packaging operation, the control inspector and packaging supervisor must check and verify the line. It should be thoroughly clean and completely free from all materials from the previous packaging order. Further, various pieces of packaging equipment are stripped down, cleaned, and examined when an order is finished. Qualified packaging personnel, with the cooperation of a control inspector, checks, endorses, and dates the bulk of the product and each of the packaging components. Practically, only the exact number of labels as per requisition for a batch, including a small axis, should be delivered to the labeling area. The packaging operation should be performed with adequate physical segregation of products. Proper online inspection should be made during the packaging operation to ensure that no foreign drugs and labels are present. The containers and closures are adequate and labeling is accurate. At the end of the packaging operation, the unused and wasted labels should be properly reconciled and disposed of. The yield must be justified against the theory represented by batch size of the starting materials. Suitable and reasonable procedures should be established for action to be taken when an unexplained mismatch is there between the number of labels issued and the number accounted for on the finished product. Key personnel should prevent distribution of the batch concerned and of other batches of products that were packaged during the same period of time until a satisfactory explanation for discrepancies obtained. Now, validation under GMP. Qualification is generally related to equipment and is used to determine whether the equipment operates as it was designed to in a reproducible manner or not. Validation of a process is the demonstration of the fact that controlling the critical steps of a process results in products of repeatable attributes or causes a reproducible event. The customary sample size in end product testing does not provide sufficient statistical validity for high product quality. It alone cannot verify that the various factors in the system intended to assure quality within and between batches of product are functioning as they were designed to function. Such verification can only be accomplished through identifying the critical components of the system and implementing control tests for these components, which in totality demonstrate that their characteristics are repeatable from batch to batch of the product. Consequently, a validated process is a systematic, documented program that provides a high degree of assurance that a specific process will consistently produce a product meeting its predetermined specifications and quality attributes. FDA defines a validated manufacturing process as the one that has been proved to do what it purports or is represented to do. The proof of validation is obtained through collection and evaluation of data right from the process development phase leading to production phase. Validation necessarily includes process qualification as well as control of the entire processes for repeated batches or runs. Now, control and assurance of finished products under GMP. Merely GMP's well-defined specifications, sound sampling procedures, and efficient process controls do not constitute an overall quality control program. Its adequacy is equally important, which is dependent on the equally sound testing procedures set up for final measurement of product quality. Product quality assurance is not complete with the release of the batch. Therefore, the testing of the finished products for compliance with the established standards prior to the release of the material for distribution is a critical factor for quality control and assurance. The testing indicates the possible deviations from perfection that occur in the batch. Product failures causing rejections or recalls after market introduction are serious and can be easily detected and minimized by an effectively administered quality testing program. However, the stability of the product in the marketed package should be repeatedly reconfirmed by actual physical, chemical, and biological tests performed on several representative batches of the product over the period of its expected shelf life. The activities associated with the control of the dosage form after manufacture will be discussed now. First of all, we will be discussing specification development. Specification writing for pharma industry is quite unique due to the element of potential hazard to public health involved and the risk of violating stringent and exacting statutes. Specifications are developed to ascertain critical, important, helpful, not particularly important but useful characteristics of dosage form. These decide dosage form characteristics to be established as the criteria for evaluating routine production batches. These also establish the appropriate test methods for evaluation of selected criteria and determine the acceptable tolerances and limits for each dosage form characteristic. Specifications are set up to ensure confirmation of characteristics of finished dosage forms to appropriate standards of identity, quality, purity, potency, safety, and efficacy. Identity, quality, purity, and potency are distinctively analytical in nature and are referred to as drug standards. Identity tests are usually the distinctive qualitative chemical methods used to confirm the actual presence of the compound. For some drugs, microbiological and pharmacological tests also may be employed. Quality tests are usually the physical methods used to measure accurately the characteristic properties of the drug. Results are expressed as a permissible range of values for a measured property of drug. Purity tests are generally designed to estimate the levels of all known and significant impurities and contaminants in the drug being evaluated. These standards are numerically expressed as maximum tolerable limit or the absence of an impurity or contaminant based on the prescribed methods. Potency tests are assays that estimate the quantity of active ingredient in the drug, employing physical, chemical, biological, pharmacological, or microbiological means. These quantitative tests yield the strength or potency of the drug substance. For drug substances, most of the chemically synthesized raw materials have a lower specification limit of at least 97% potency, with the exception of antibiotics. However, different dosage forms of the same drug substance may be assigned different specification limits as well. Positive and negative specification limits are generally symmetric around the potency for most of the dosage forms. Therefore, in many cases, potency range is expressed in the form of 100 plus minus 10% or 100 plus minus 7%, then 90 to 110% or 93 to 107%. A drug substance is suitable for use if totality of data derived from these four attributes shows that it meets all of the specifications. Generally, specifications make provision for maintenance of reasonable standards. Further, they allow a sensible degree of latitude for manufacturing variations and acceptable latitude for analytical errors, particularly in control techniques. In the appropriate development of specifications, historical data accumulated from batch analysis of the drug product are usually the best source of guidance. Specifications are also used in the procurement of drugs, excipients, reagents, packaging, and printing materials. The incoming materials and finished products are checked against specifications to determine their compliance and acceptability. Specifications for purchased materials should be developed based on the vendor's capability to supply the material. Specifications should be practical, realistic, and reflective of parameters necessary to define both the product and its quality level. No single set of requirements meets all possible situations. Now, specifications for new products. During the development of new drugs, both their chemical and physical properties should be rigorously investigated by nuclear magnetic resonance, infrared spectroscopy, and mass spectrometry. This testing usually provides strong evidence of structure and hints at identity of possible byproducts that might be present. Specific chemical reactions may give further confirmation of structure. Generally, Generally, possible contaminants are structurally closely related to drugs and might have an undesirable or even harmful effect. Thus, it is reasonable that its presence should be defined and controlled. 2 to 3% of such substances may not be readily detectable even by infrared spectroscopy. It is necessary to recognize the presence of impurities present at such low levels. Although ideally a drug should be absolutely pure, this is generally not feasible in practice. A more practical approach is to require that material to be subjected to pharmacological and clinical trials is prepared to be as pure as is economically reasonable and then characterized as fully as possible with respect to its impurity content. If the trials prove satisfactory and acceptable, then it should be ensured that the subsequent batches of material should be at least as clean as the trial material. It is essential that total identity of material being used, including both its major and minor constituents, should be known. Only in this way can the results of clinical trials be meaningful for a new drug. Now, specifications for well-established products. The material synthesized on a pilot scale for clinical trial work may not be the same as material synthesized in a full-scale manufacturing process, especially with respect to its pattern of minor constituents. Every impurity in each drug must be considered as a separate and individual case. If the impurity has been shown to have or is suspected to have undesirable properties, it should be limited as rigorously as possible. Lastly, official specifications under GMP. The specifications of the manufacturer are designed to be applied to the drug at the time of manufacture or quite shortly thereafter. An official specification, on the other hand, should be designed to apply to the material at any time during its period of possible use. An official specification is likely to be somewhat less stringent, especially with regard to changes due to slight decomposition. Specifications for a product are developed not only to assure product quality but also to detect and identify impurities. There are two major kinds of impurities. First kind is product non-specific impurities, which are introduced externally into the product during processing, and the second kind, product specific impurities, which appear as byproducts or degraded products of the drug substances as well as excipients used in the product. Therefore, the test parameters should be designed and testing procedures for the specifications should be established so that the tests are capable of detecting product specific as well as product non-specific impurities. A description of the test and reagents for various official drugs and their dosage forms are available from official compendia. Tests are mandatory for proper control of the product. All drug specifications listed in pharmacopoeias have been verified in collaborating laboratories of pharmaceutical companies and academic institutions. Pharmacopoeia issued for a country is the legal standard of that nation. Specifications in a pharmacopoeia are so designed that tests and requirements for acceptance are applicable to all manufactured products. This means that anyone who manufactures a product of that type should conform with those specifications. It should be recognized that the specifications set in the compendium are minimum standards to which the product is expected to conform at any time during its expiry date period. A compendium is a collection of monographs of drug substance and drug products. Official, as used in official compendium, signifies governmental authorization. Procedures in the compendium apply to all manufacturers of particular product, whereas the quality control procedures of manufacturer are intended to apply specifically to his own product. The pharmaceutical industry frequently employs alternative methods which may be more accurate, specific, sensitive and economical than those in the compendium. The manufacturer is not required to employ procedures in the official compendium as long as the quality of his product ultimately meets the requirements in the compendium for identity, quality, potency and purity. In the case of a legal action, test methods in the compendium are the basis for determining compliance. Container components should not interact physically or chemically with the product to alter its identity, purity, quality or potency beyond the official allowances. Usually, the major component of the container is glass or plastic. The official compendium provides for several types of glass and several classes of plastic to be used with various pharmaceutical products. Specifications designed for containers are meaningful only if they have been selected on the basis of tests performed on the product in the container. Certain features are to be considered before container specification, which include physical changes of container upon prolonged contact with the product, moisture and gaseous permeability of the container, compatibility between container and product, and toxicity and safety. So students, we had a good introduction to good manufacturing practices. We discussed about consequences of GMP compliance. We also talked about GMP guidelines, GMP certification, and lastly, we discussed at length personnel, equipment, building, control of production records and procedures, and elements of manufacturing control under GMP. Thank you.